All right, hi everyone. I'm going to go through in more detail the DNA replication process with you. And I just wanted to cover a couple things at the beginning regarding the structure of DNA first. So DNA is called deoxyribonucleic acid. Each piece of this is going to give away part of its component. So the deoxyribo part is referring to right here. That's referring to the sugar um, that is attached to it because DNA and RNA are always compared to each other. They are very similar with their backbone and their structure. And so the deoxyribo is the sugar that goes with DNA, whereas RNA has a ribose sugar, which is it's called ribonucleic acid. And then we should know that nucleic acids are going to be made up of three components anyway. We already talked about and referenced the sugar. That's one of them. The phosphate is the next one. So that's our PO4 component. And then the base. So whether that's A's, T's, C's, or G's. And again, you don't have to know the actual name of these. You just have to know the letters. So again, ribose sugar is going to be over here. If we're looking at our diagram, here's our sugar, all these orange molecules that are our 5-carbon sugars, right? These are part of the backbone of DNA. So this gives it its solid structure. It's going to have a covalent bond that is attached to it. And it's going to bond with, we can see these little dots over here. So here are all my covalent bonds that are happening. And then it's going to bond with this little dot over here, and that's going to represent our PO4 or our phosphate molecule. So phosphates and sugar are the backbone, the solid structure that holds DNA together. And then the bases are pretty interchangeable, just a couple different types that go along with that, but we can see that there's A for adenine, G for guanine, T for thymine, and C for cytosine. And there's these dashed bonds between them. Those are our hydrogen bonds. We know hydrogen bonds are weak, they can separate, and that's an important component for DNA replication so that it can separate and reform really with ease. So those hydrogen bonds go between their bases. We should also recognize our base pairing process of A's pairing with T's, G's pairing with C's, always, always, always. And then we can see the same thing on this side with our backbone of sugar, phosphate, alternating back and forth. Okay, let's see. I'm going to just say that does not sound like a very fun job, at least in my opinion. If I move on, I knew that was going to be there. Sorry about that. So here is our central dogma, the key component to biology, our process. We're going to focus on the DNA replication step first. So we're going to talk about all the different enzymes that are involved in how we get a new strand of DNA. That's basically the exact same, just written in kind of in the opposite order, based off of our base pairing rules or our complementary base pairing rules. So here we go. It goes through a process called semi-conservative replication, which basically means we're going to take an original parent strand of DNA. Normally they're wound up like this. They will separate and unwind, and then in the end, we'll have one original parent strand and a new one that is formed. So I'll say this is my new strand that's going to form over here. And then on the other side, here's my original parent, and then a new strand is going to form based off of those DNA bases and pair them up. So that's semi-conservative when there's an original template strand with a new one. It's not just new or original parents over here and new template strands over here. It's always going to be one new and one old next to each other. Here are our key enzymes, all ending with ACE. Each name is going to kind of give a little hint as to what it's acting on. So my first one is called helicase, and that's going to act on the double helix of DNA. So helicase goes through and attaches and unwinds and unzips DNA so that we have our two strands that we can kind of work with. It creates a replication fork, a fork in the road, so that it can separate and unwind. So if I were helicase, I'd want to unzip your genes. That's what the dirty joke is about helicase. So it unwinds and unzips the DNA strands and creates a replication fork. DNA primase will attach to one of those strands and lay down, an RNA primer. So RNA is going to have A's, U's, C's, and G's attached to it. This RNA primer is important because it's going to help identify the starting point for where the bases should be added and the new strand can be made. So helicase unwinds and zips, creates a replication fork. RNA DNA primer creates an RNA strand of about 10 to 20 bases that hints, hey, here's where we're starting replication. Topoisomerase is going to attach up here at the replication fork, and it's going to help alleviate any of the stresses from this unwinding and unzipping. So usually there's a big ball or a jumble that would happen up here because of all the tension of stretching and pulling DNA apart. Topoisomerase will kind of lengthen and stretch and separate and relieve that tension. DNA polymerase has a couple components to it. There's actually 3 types of DNA polymerase out there. DNA polymerase 3 is our main component polymerase because it is going to be making the long chain of new DNA and the new strand that is going to be based off of our complementary base pairing rules. So DNA polymerase 3 does this. It finds that RNA primer that was laid down. It reads it, says, okay, I'm attaching here next to the RNA primer, and I'm going to start making my base pairs complementary to what I'm reading. So if there's a C, I'm laying down a G. If there's an A, I'm putting down a T. So on and so forth, all the way through. And then ligase is the glue of DNA. And once those bases have been proofread by DNA polymerase 1 does the proofreading. It'll read it, make sure, and then ligase will go through and glue it all together. So we have one nice new strand of DNA that has paired up with the old one. They'll eventually wind up again. So here's that visual of it. Let me get my head out of the way. Here's our helicase. Step 1, unwind, unzip, create a replication fork. We got our primase, which is going to lay down an RNA primer and allow it to be the original code so that DNA can be made. We got our topoisomerase up here at the top towards the replication fork where it's going to stretch and alleviate any tension that's happening up there. And then we got our DNA polymerase 3 that's going to go through and attach to that RNA primer and start adding new bases after that. We can see that there's a couple different steps. The leading strand up here is always going to be read in one clean, continuous motion with DNA polymerase and create this long strand uninterrupted. Down here on the lagging strand, it's going to go in little chunks, and RNA primer goes on the lagging strand, and it'll be in several different locations. So DNA polymerase 3 will create a little chunk of DNA, find the next RNA primer, create another chunk, find the next RNA primer, create another little chunk until that whole lagging strand is developed. So that'll go through. Here's our DNA polymerase 1 over here our DNA polymerase 1 over here that is going to go through and do some, oh boy. I really messed that up. That's going to do some proofreading, and it goes through a process called exonuclease, where if there's something wrong with DNA, it's going to go through and edit it and make sure that it is the correct base pair that's going on. So DNA polymerase 1 does the proofreading and double checking, and then ligase comes through and seals all the new bases together. DNA polymerase 1 also goes through and kicks out that RNA primer that was laid down and replaces it with actual DNA bases. I have this one linked. It's about a minute long to our original note section because this one is not the proper one. Sorry about that. But it goes through and shows you DNA helicase and how it's the main player in creating the replication fork. How do I know whether I'm working with the leading strand or the lagging strand of DNA? Visually, if you can see that one new strand is being made in a continuous motion, you know you're dealing with the leading strand, and the lagging strand goes in little chunks, which I'll talk about next. So that's one way of visually seeing it. It's also going to be notified or noted in a 3' and 5' location. And so the leading strand is always going to be going 5' to 3', and the lagging strand is always going to be going 3' to 5'. This just is called anti-parallel, which means although we're putting in complementary bases, we're going to end up with the same sequence of DNA. It's just going to be going in the opposite direction. So after DNA unwinds and zips, we identify our leading and our lagging strands. We have our primers, and our polymerase is going to come and attach in phase 2, where our new complementary chain of DNA is going to be added. So that's our step 2, is assembling new complementary chains. Get my face out of there. Again, using the semi-conservative method, there's always going to be one old strand with one new strand on both sides, so that in the end we have one old and one new next to each other. It uses those complementary bases. We can kind of see them down here: A's with T's, C's with G's, C's with G's, A's with T's. And that's how the new strand is made. DNA polymerase 3 does the adding of the complementary bases, right? Again, leading versus lagging strand. How do I know? Leading strand goes, here I am in the way again, goes 5' to 3', that direction. And then lagging strand is 3' to 5', or the top. And we can see that based off of the structure, whether or not there is a phosphate hanging off at the end, or if it's a sugar from the backbone that's hanging off from the end. So that's one way to identify it. Anyway, here's our leading strand. That's going to create a new complementary strand in one swoop, uninterrupted by that DNA polymerase 3, whereas the lagging strand goes in little chunks. So the lagging strand is going to have multiple primers attached to it. Polymerase will attach, create new bases in those little chunks until it encounters a new primer, reattach, create new bases, reattach, create new bases, and then it'll go back, and our DNA is going to continue to unwind and unzip. So then there's a new primer up here, and it's going to add new bases and create it in little chunks. This avoids mutations and improper complementary base pairing. And those are called Okazaki fragments, those little chunks of DNA that are made on the lagging strand as the replication fork opens up. I said before, DNA polymerase has kind of a multiple roles. So DNA polymerase 1 is actually going to go through and do some proofreading, check for errors, and then make some corrections if there's anything wrong. And that's called enzyme nuclease, or a nucleotide excision repair process. Yeah. So if there is an A paired with a C, like we see down here, I'll kind of let that reform. It'll go through and kind of take an axe and chop out the old base. It'll find a new one and say G actually goes here, and plop that into place. It will bring in ligase and seal it in the proper location. And so if there's any damage or whatever, DNA polymerase 1 does the corrections and the changes. So, step 1, unwind, unzip with DNA helicase. Create the replication fork. RNA primase lays down the primer. DNA primase lays down the RNA primer. There's 3 over here on the lagging strand. Okay. Our DNA polymerase in step 2 is going to create a long chain on the leading strand in one continuous motion. On the lagging strand, it goes through and reads in little chunks called Okazaki fragments. Ligase will come through and seal it up if everything's correct. Otherwise, DNA polymerase will go through and chop out the bad bases and replace them with the good ones. And then it's all good. So that's our DNA replication in 3 lovely steps.